Today we are going to discuss Krebs cycle. It is also known as citric acid cycle. Why it is called citric acid cycle? Why Krebs cycle is called citric acid cycle? It is called citric acid cycle because the first product in the Krebs cycle is citric acid, citrate. When oxaloacetate is react with acetate, it will form citrate, citric acid. So it is called citric acid cycle. How we can define Krebs cycle? How we can define? This is a chain of reaction. This is a chain of reactions. Which type of reactions? The decarboxylation and dehydrogenation reactions, in which the energy is stored in the coenzymes like NADH and FAD. This NAD and FAD are rechargeable batteries in the cell. It will recharge in the Krebs cycle and goes to electron transport chain for energy release. Or we have two types of money. One is in cash form and one is in check form. For check we are going to bank and cash is cash. So the ATPs directly producing in the Krebs cycle or in the glycolysis are the examples of cash. This is cash energy, direct energy. And the NADH and FADH are like a check. We will go to the bank of the cell, and the bank of the cell is electron transport chain, and we will cash the check, right? So this is the series of reaction. Why it is called cycle? Why Krebs cycle is called cycle? Glycolysis is not cycle. It is because the first product is just the same as the end product. The oxaloacetate is the first product of the Krebs cycle, which react with the acetyl coenzyme A and form citrate, and in last from citrate it will be again converted into oxaloacetate. So it is called cycle. Which reaction, which pathway is called cycle? When the first and last product are the same. This is called cycle. What is the location of Krebs cycle? Where Krebs cycles take place? Mitochondria, the matrix of mitochondria, right? So for example, I have a cell. I have cell, and we have discussed in detail that in cytoplasm glucose will convert into 2,3 carbon compound which are called pyruvates, right? This is the process which is take place in cytosol or cytoplasm, right? And here is a very important cell organelle which is called mitochondria, and mitochondria is double membrane structure, right? For example, this is mitochondria, and this area is called matrix of mitochondria. Matrix of mitochondria. This area is called intermembrane space. Intermembrane space. There are two types of membrane. One is this is called outer mitochondrial membrane, and this membrane is called inner mitochondrial membrane. Now, what are the unique characteristics of this outer membrane mitochondrial? This is very, very permeable to a lot of things. It can allow the things from outside to inside mitochondria. But this inner membrane is so much impermeable, semi-permeable. It will allow only the needy substances to enter into the matrix of mitochondria, right? So when this pyruvate is formed in this, for example, this is 3 carbon compound. When this pyruvate is formed in cytoplasm, it will enter into the matrix of mitochondria, right? And here it will convert into acetyl coenzyme A. But strictly remember this point with yourself, that the formation of acetylcholine is not the step of Krebs cycle, and it is also not the step or the process of glycolysis. This is the link between glycolysis and Krebs cycle, and that's why it is called link reaction. It is called link reaction because when link reaction is occurring in the mitochondrial matrix, there will be the formation of NADH, which is the stored form of energy. Also from here, why this Krebs cycle is called the decarboxylation series of reaction, right? If you remember the whole reaction of the breakdown of glucose, C6 H12O6, when there is abundant oxygen, it will convert into 6 CO2 plus 6 H2O plus 38 ATPs. It is in the form of aerobic respiration when there is abundantly oxygen is present in the cell, or the mitochondria is present in the cell, because we have a cell in the body which doesn't have mitochondria, and the cells are RBCs, erythrocytes, right? So here you can see 6 carbon dioxides are released from this glucose, and how many carbons are here? 6. It means that in each carbon dioxide 1 carbon is from glucose is excreted, right? For example, this is 1 2 3 4 5 6. 6 carbons are here. This is called carbon skeleton, right? So for example, this carbon is excreted. This is called decarboxylation reaction, and on the way two oxygen will react with this and will form carbon dioxide. From here also carbon dioxide, carbon dioxide, carbon dioxide, carbon dioxide, and carbon dioxide. So 6 carbon dioxides are formed in this reaction. From where the carbon is coming? The carbon is coming from glucose, right? But when there is decarboxylation reaction, because these carbons are attached to hydrogens and hydroxyl groups, right? So for example, from here 1 hydrogen is excreted and 1 hydroxyl group, it will form water. It will form water. This is called the dehydrogenation reaction, right? So this Krebs cycle is called the series of decarboxylation as well as the dehydrogenation reactions, in which the energy is stored in the form of coenzyme FADH2 and NADH, right? This is the full definition of Krebs cycle, and it will take place when there is aerobic respiration, when there is abundantly oxygen is present. If oxygen is not present, Krebs cycle will not occurring in the cell or the mitochondria, and the pyruvate will go for the fermentation reaction. Lactic acid will form, right? And lactic acid formation is, in some cases, lactic acid formation is beneficial in moderate or the enough amount. But when the lactic acid is forming in very high amount, it will cause the lactic acidosis. Lactic acidosis is very dangerous. It have 3 processes, it have 3 types: a very severe lactic acidosis, the moderate lactic acidosis, and mild lactic acidosis. We will discuss these 3 types in the end of this reaction, right? So this is some of the introduction to Krebs cycle. Now the process we have discussed last time is glycolysis. We will quickly review this process. For example, we have 6 carbon compound which is called glucose, right? In energy consuming phase, it will convert into 1,6 fructose 1,6 base phosphate, right? And here how many energy is used? 2 ATPs. Remember this with yourself, right? We are not going for again discussing the enzymes. This is the process, this is energy consuming phase in which the glucose, the 6 carbon compound, is modified or converted into fructose 1,6 base phosphate, and 2 energy are 2 ATPs are used here, right? The next is splitting phase. In splitting phase, this molecule is converted into 3 carbon compounds. It will convert into 3 carbon compound. One is called dihydroxy acetone phosphate, and second is called glyceraldehyde 3 phosphate. These are interconvertible, right? Now this will convert into glyceraldehyde 3 phosphate, glyceraldehyde 3 phosphate, and after it will convert into pyruvate. Series of reactions, after undergoing series of reactions, it will convert into pyruvate, and it will also convert into pyruvate. So from one glucose molecule, 3 carbon compound pyruvates are formed, right? But during this reaction, one NAD is converted into NADH. Here one ADP is converted into ATP, and another ADP is converted into ATP, right? Here also one NAD is converted into NADH, one ADP is converted into ATP, and here also one ADP is converted into ATP, right? This step is called energy production phase, right? Here how many ATPs directly? This is the cache energy, this is the cache energy, which are substrate level phosphorylation. Any ATP production which doesn't need oxygen is called substrate level phosphorylation, and these are doesn't want oxygen, so all these cache energy is formed by substrate level phosphorylation, right? So calculate all the ATPs here. 1, 2, 3, 4. 4 ATPs are directly formed. 4 ATPs. Right? In which phase? In energy consuming, energy production phase. One is check. One is in the form of check, NADH. This NADH, of course, we know that when it is passing from electron transport chain, it will equal to 3 ATPs, right? Here we have 2, 1 and 2. We have 2 NADH, so it is equal to 6 ATPs, right? This process occur when there is abundantly oxygen is present. If oxygen is not present, it will not undergo the electron transport chain, right? Here how many energy is used? 1 and 2. So the gross, it is 10 ATPs. We will subtract this 2 ATPs from the gross, and we will get the net ATPs, which is 8 ATPs. This is why I am discussing this. It is important, right? It is very important when we will calculate all the energy production in the aerobic respiration, right? Here is the game is different. when there is oxygen, when oxygen is present abundantly, it will undergo Krebs cycle. It will undergo Krebs cycle, and it also undergoes Krebs cycle. Mean for one glucose molecule, how many Krebs cycle will run? 2, because 2 pyruvates are formed here when there is enough oxygen is present, right? Here, this reaction, I will explain this reaction today. When this pyruvate is converted into acetyl coenzyme A, and energy will produce. Now listen. Here, when this pyruvate is converted into acetyl coenzyme A, acetyl CoA, acetyl CoA. Here, 1 NAD is converted into 1 NADH. Right, here also 1 NAD is converted into NADH. So how many ATPs are formed here in link reaction? 6 ATPs. In link reaction we will discuss 6 ATPs that are formed, but here this reaction occurs in 3 steps. One is decarboxylation reaction, mean 1 carbon dioxide will released. Here also 1 carbon dioxide is released. This is the first carbon dioxide which is released in this process, right? And the 2 other carbon dioxide will release in 1 Krebs cycle, 2 and release in next Krebs cycle. So the net will be 6, right? How many NADH will formed in Krebs cycle? In Krebs cycle, in 1 Krebs cycle, 3. Here 3 NADH. We will discuss this in detail, how 3 NADH are formed here, right? 1 FADH2 is formed, and here 1 substrate level phosphorylation take place. 1 GDP will convert it into 1 GTP. Guanosine diphosphate will convert into guanosine triphosphate, and this guanosine triphosphate will give its inorganic phosphate to ADP and will convert ADP into ATP. So 1 cache ATP is formed here, right? Now 1 NADH is equal to 3 ATPs, so 3 will be equal to 9 ATPs, right? Here 1 FADH is equal to 2 ATPs, so 1 FADH it will give us 2 ATPs, right? And 1 ATP, so it will equal to 1 ATP. Now calculate it: 9, 10, 11, 12. If you have an MCQ in your paper that how many ATPs are formed in 1 Krebs cycle, students are always confused in this type of questions, right? When they are asking about 1 Krebs cycle, the net ATP in 1 Krebs cycle is 12. If they are asking about full glucose, the net ATP will be 38. If they are asking about 2 Krebs cycles for 1 glucose molecule, this energy will be 24. This energy will be 24, because here from this Krebs cycle also 12 ATPs are formed. For example, I am giving you a question: how many ATPs are formed in Krebs cycle? 12, 24, 8, 38. 12. 12. If they are asking how many ATPs are formed in Krebs cycle for 1 glucose molecule, 24, right? So 8 ATPs are here and 12 are there, right? How many they are now? 20. Up to now we have 20. Here in link reaction, we have 2 NADH and 1 NADH is equal to 3 ATPs. So 3 ATPs and 3 ATPs, it will gives us 6 ATPs. It will give us 6 ATPs, right? So now 12, 24, 6 and 8. Full 2 Kreb cycle will give us 24 ATPs. It is 8 ATPs and 6 ATPs. Now calculate 24 plus 8, 32 plus 6, 38. So the net ATP production for one glucose molecule when there is oxygen, when there is enough oxygen, is 38 ATPs. And when there is no oxygen, or lack of oxygen, only 2 ATPs will form from glucose molecule with the formation of lactic acids, right? So this is the story behind ATP production of glucose. We will calculate this also in last of when we will discuss electron transport chain, right? Because these NADH and FADH well converted into ATPs in electron transport chain, right? But keep it in your mind, right? Now we will start the Krebs cycle. For example, I have a mitochondria here. Do you know the Tem and Tom protein? Tem and Tom, non Tom and Jerry. Tem, Tom, or Tom and 10 protein. These are very important proteins of mitochondria, present in mitochondria, right? So for example, I am taking one mitochondria here. For example, what is the shape of mitochondria? It may be spherical, it may be cylindrical, it may be cubic, it may be rectangular, right? So this is the, for example, this is the mitochondria. I am taking only mitochondria, not the whole cell. And mitochondria have how many membranes? 2. One is inner and one is outer, and both are very important. And what is the basic difference between the outer and inner membrane? There are two differences. One is the outer membrane is smooth and the inner membrane is folded. The second difference is the outer membrane is very permeable and the inner membrane is selective permeable or semi permeable. So this is the outer membrane and this is the inner mitochondrial membrane. Right? This area is called mitochondrial matrix. This area is called intermembrane space. This black membrane is called outer mitochondrial membrane and this blue is called inner mitochondrial membrane, right? From here, in this glycolysis, the end product is pyruvate. The end product is pyruvate. So for example, I am taking pyruvate. This is pyruvate have how many carbons? 3. There is a strong bonding between these 3 carbons. right? And this molecule is the end product of glycolysis, which is pyruvate. And how many pyruvates are formed in glycolysis? 2. So there will be 2 pyruvates, and this pathway will run 2 times, right? Why this Krebs cycle is undergoing in occurring in this matrix? Because all the enzymes which are responsible for catalyzing this Krebs cycles are present in the matrix of mitochondria. If the cell doesn't have mitochondria, the cell will not able to run Krebs cycle like erythrocyte or RBCs, right? So this is pyruvate. This pyruvate can easily pass from outer membrane through which protein? The Tom protein, right? Here are the proteins. For example, this is the protein, and this is called Tom. Tom stands for transferase of outer membrane. Transferase of or transporters of outer membrane. Tom, right? It will take this pyruvate from outside to inside the outer mitochondrial membrane, right? Remember, pyruvate is 3 carbon compound, right? From here, this pyruvate will taken by the 10 protein. Here, for example, we have this is the 10 protein. This pyruvate is taken by this 10 protein and allow it into mitochondrial matrix. So pyruvate is now present here with these 3 friends. They are coming together here. They are happy because they don't know what are happening to this friendship, right? This is pyruvate. Three carbon compound. In mitochondrial matrix, this pyruvate will convert into acetyl coenzyme A, but this is not the step of Krebs cycle. This is not the step of Krebs cycle. So this pyruvate, for example, this is the reaction. It will convert remember, when I am making the step like this in green, this step will be very regulated and well irreversible. This is irreversible step. This pyruvate can convert it into acetyl coenzyme A, but this acetyl coenzyme A cannot revert into pyruvate, right? So this is the most regulatory step in this process, right? What are the regulatory steps in glycolysis? The first one is when glucose is converting into glucose 6-phosphate. Second step is when fructose 6-phosphate is converting into fructose 1,6 base phosphate. And the next is when phosphoenol pyruvate is converted into pyruvate. These 3 steps are very high regulated steps. And here this link reaction is also very important, right? What will happen to this pyruvate? It will convert into, now they are unhappy because they lose one friend in the form of carbon dioxide, right? First carbon dioxide is formed in this step, right? From these 3 carbons, 1 carbon will convert it into carbon dioxide and the 2 carbons are remaining here, which is called acetate or acetyl. and in this reaction one coenzyme A group is added to coenzyme A. It is added to this product, and now this product is called acetyl CoA, and this is now the intermediate molecule which is responsible for undergoing Krebs cycle. Not only the glucose will converted into acetyl coenzyme A, but also the proteins and lipids will also convert into proteins and lipids will also convert into first acetyl coenzyme A, and this coenzyme A will run Krebs cycle for energy production. The molecule which have more carbons will give us more energy, and the molecules which have less carbons will give us less energy. Alright, this is the clear example, the comparison between glucose and carbohydrates and fats. The fats have a lot of carbons, and fat is 1 gram of fat is responsible for producing 109 ATPs, and one glucose molecule is responsible for making 38 ATPs. There is a lot of difference, right? So here this step, why? We are focusing on this step which is very regulatory. 3 types of reactions are taking place with this, or 3 types of tragedies this molecule will face. The first tragedy is it will lose 1 carbon dioxide. It will lose 1 carbon. This is the first tragedy, and this reaction is called decarboxylation reaction. It is called decarboxylation reaction. Right, decarboxylation reaction. The second tragedy, the second one, here acetyl coenzyme A is added. Not acetyl, only the coenzyme A is added to this reaction, and this is called coenzyme A binding reaction. Coenzyme A binding reaction. This is the second. The third one is 1NAD. 1NAD plus is added to this reaction, and when it out from this reaction it will become NADH. There are 3 reactions take place in this link reaction. This is called this reaction is called the production of coenzyme, or this is called reduction. This is called reduction because hydrogen is added, right? But in other way, this is the dehydrogenation reaction. How? For example, from carbon the 1 hydrogen is released, and this the pyruvate is dehydrogenized, and this NADH reduced, right? So this reaction is called dehydrogenation reaction, right? And in start I have told you the Krebs cycle is the series of the decarboxylation and dehydrogenation reaction in which NAD or the energy is stored in the form of NADH, or NAD is reduced into NADH, right? This is the stored form of energy. Now we will discuss how these reactions take place and how this reaction, this link reaction, is regulated. Regulation is very important because there are a lot of clinical conditions which are dependent upon only this reaction, right? For example, if a person have a loss of appetite, anorexia, this is due to this reaction, and this reaction is stopped, you will stop eating because metabolism is stopped, Krebs cycle is stopped, energy production is stopped, inhibited, right? Other, lactic acidosis. Lactic acidosis is inherited disease which is coming from this reaction, right? Because if there is some mutations or some abnormalities occur in this enzymes or in this reaction, it will affect our body, right? What is that lucky enzyme which is working in this important reaction? It is called pyruvate dehydrogenase complex. The enzyme which is working in this reaction is pyruvate dehydrogenase complex. Why it is called complex? It means that there are some units which are combining and forming this enzyme, and initially it have three components. This enzyme, the pyruvate dehydrogenase, have three components: E1, E2 and E3. E1 is composed of thymine pyrophosphate. E2 is composed of lipoic acid and coenzyme A. E3 is composed of NAD plus and FAD plus, right? And the first E1 enzyme is called pyruvate dehydrogenase enzyme. The second, the E2, is called dialipolyl decarboxylase enzyme, and the last one is called dialipoic synthase enzyme or dehydrogenase enzyme, right? These E1, E2 and E3 are responsible for regulating these three reactions. There are three reactions and three enzymes, so it is called pyruvate dehydrogenase reaction. Also, two components are responsible for regulating this step. One is phosphatase enzyme and the second enzyme is kinase enzyme. Phosphatase and kinase enzyme. The kinase is very cooperative, and this phosphatase is very anger, right? This phosphatase shutting down this enzyme, this pyruvate dehydrogenase, and kinase enzyme came and regain this enzyme or rephosphorylate this enzyme for working. So now we will discuss this whole process, how pyruvate is converted into acetyl coenzyme A, how this carbon dioxide is released from here, and how this NAD plus is converted into NADH, and how this coenzyme A is working, and how this step is dependent upon thymine. What is thymine? This is B1 vitamin. Thymine is vitamin B1, and vitamin B1 have a very important role in this reaction, right? And in last, we will discuss the clinical points. Only three clinical points or four. One is very severe lactic acidosis. The second is moderate lactic acidosis. The third is mild lactic. The worst one is mild lactic acidosis. And first two steps, you will know why intrauterine deaths occur when the baby is die in uterus. Why intrauterine deaths occur? Because of lactic acidosis. Why the deaths occur in infancy? When they are delivered, and after delivery they die. Why they are die? Because of lactic acidosis, because they have a mutation or they have abnormality in the genes. There's heredity, right? So we will discuss all these mechanism, all these clinical points, all this pathway, right, and very big detail, right? And any question up to here? These are पूरे सब question in okay. And this acetyl coenzyme A, when there is abundantly oxygen is present, this acetyl coenzyme A will undergo Krebs cycle, right? And Krebs cycle, there will be release of two more carbon dioxide. These are decarboxylation reaction. There will be dehydrogenation reaction. For example, here one NAD will reduce into NADH. Here also one NAD is reduced into 1 NADH. Here 1 GDP is converted into GTP, and this GTP converts 1 ADP into ATP, and here 1 FAD is converted into FADH2. Right, so this is the series of decarboxylation as well as dehydrogenation reactions which occur in mitochondrial matrix. What is the purpose of Krebs cycle? It stores the energy in the form of coenzyme which is called NADH and FADH2. Right, so this is the whole process of Krebs cycle, and if there is oxygen, this will take place. This is for 1-acetyl coenzyme A, and this process will occur twice because there will 2 pyruvates. So I have told you 12 ATPs will formed here, and 12 ATPs will form in another cycle, and the net energy production for 1 pyruvate molecule is 12 ATPs, and for 2 pyruvate molecule is 24 ATP. So this is the process, right? We will discuss this Krebs cycle in detail, but first we will discuss this, and this reaction, especially this reaction is called link reaction. सही शो पोई पदे वांदे कसा क्वेश्चन ही माना ओके निष्ठा क्वेश्चन बिल्कुल निष्ठा पास 5 मिनट ब्रेक ओके शावेनारानवल. This is mitochondria. Mitochondria have 3 or 4 important parts. One is outer mitochondrial membrane, one is inner mitochondrial membrane, one is inter membrane space, and one is matrix. Right, why I am mentioning all these parts? Why? Because this outer membrane is more permeable. Remember this point and keep it in your mind for future. This outer membrane is highly permeable. This is very selective permeable, and this is smooth and this is folded. Right, intermembrane space. What is the importance of intermembrane space? And last, when we will discuss electron transport chain, all the hydrogen ions will accommodate in intermembrane space, and when these hydrogen ions will go inside, it will form energy by rotating the ATP synthase enzyme. Right, and matrix, mitochondrial, but also in intermembrane space, the enzymes for electron transport chain are present. Here the enzymes responsible for undergoing electron transport chain are present in inner membrane of mitochondria. This matrix, all the enzymes responsible for running Krebs cycle are present in matrix. So this is the location for Krebs cycle, and this is the location for electron transport chain. These are also 2 MCQs. The location of electron transport chain is intermitochondrial membrane or inner mitochondrial membrane. The location of glycolysis is cytosol. The location of or the site of Krebs cycle is mitochondrial matrix. Right, now there are 2 very important proteins I have told you. One is TOM. This is TOM, transferase enzyme or transferase for outer membrane, transferase of outer membrane. Right, it will take the pyruvate. From where pyruvate is coming? from glycolysis. The end product of glycolysis is pyruvate. So pyruvate from here, this is for example pyruvate, and pyruvate is 3 carbon compound. So this is 3 carbon compound. This Tom protein is responsible for taking this pyruvate into the intermembrane space, and from here another protein is responsible for taking this pyruvate. For example, this pyruvate is now in intermembrane space. This 10 protein will take this pyruvate into mitochondrial matrix. Here now we have pyruvate. This is 3 carbon compound, right? But again remember with yourself that this link reaction will occur in mitochondrial matrix, but this is not the part of Krebs cycle. This is not the part of Krebs cycle from here up to here. For example, here it will convert into acetyl coenzyme A, and acetyl coenzyme A is how many carbon? It have 2 carbons. Mean on the way 1 carbon is excreted, or at this pyruvate loss 1 carbon and will convert into acetyl CoA. For example, this is acetyl CoA. Acetyl CoA is 2 carbon compound. This is 2 carbon compound, and it is attached to a side chain which is coenzyme A. This is acetyl coenzyme A. Now our topic is how this pyruvate is converting into acetyl coenzyme A, and which enzyme is responsible for catalyzing this reaction, and what are the changes which occur on the way when pyruvate is converting into acetyl coenzyme A, because without acetyl coenzyme A, Krebs cycle cannot be occur in mitochondrial matrix, right? Here this reaction is called link reaction. Why it is called link reaction? Because it joined or it linked the glycolysis with Krebs cycle. For example, here we have glycolysis, and this glycolysis, after glycolysis, for example, this is the mitochondria, it convert into pyruvate, and this pyruvate will convert into acetyl CoA, and this acetyl CoA will undergoes Krebs cycle. So this is the reaction which link the glycolysis with Krebs cycle. So it is called link reaction, right? If someone asks about link reaction, you can say that this is the reaction in between the glycolysis and Krebs cycle, because the end product of the glycolysis, which is pyruvate, cannot directly undergoes Krebs cycle, right? So here the enzyme which is used here is called pyruvate dehydrogenase. Pyruvate dehydrogenase complex. This is the enzyme which catalyzing this reaction. What are the changes occurs due to this pyruvate dehydrogenase complex? First is it will lose one carbon in the form of carbon dioxide. So one carbon dioxide is lost from here, and this is the first carbon which is lost in Krebs cycle. And this link reaction will also undergoes two times because here are two pyruvates. So two carbon dioxide will released here. The second chain, this reaction is called decarboxylation. Decarboxylation reaction, right? Second thing, a coenzyme A is added to this reaction, and it will react with the two carbon compound acetate and will form acetyl coenzyme A. Remember, in neuronal cell, this acetyl will react with choline and will form acetylcholine and the ends of neurons, right? And this acetylcholine is one of the important neurotransmitter, right? Now the third change is the NAD plus will convert into NADH. This is the stored form of energy. NADH, this is reduction reaction, or this is the dehydrogenation reaction, right? Now we will take this enzyme and we'll see how these changes occur due to this enzyme, because if you are noticing, I am focusing only on the enzyme which are highly regulated, not on the enzyme which are involved in the reversible reactions. I am only focusing on which are responsible for irreversible reaction, because the irreversible reactions or the irreversible steps in this metabolic pathways are highly regulated, right? So here, this I have told you, this enzyme have three components. For example, this is the first component of pyruvate dehydrogenase. This is the second component of pyruvate dehydrogenase, and this is the third component of pyruvate dehydrogenase. And all these complexes, or all these enzymes are working together. These enzymes are working together. So that's why this enzyme is called pyruvate dehydrogenase complex. So this is pyruvate dehydrogenase complex, right? This first component is called E1, the second component is called E2, this third component is called E3, right? So there are three components. One is enzyme 1, enzyme 2, enzyme 3. Initially this complex have three components. It is composed of three further enzymes, right? Now the first one, for example, I have the components which are very important here. The first component have one coenzyme, right? And this coenzyme is called thymine pyrophosphate. Thymine pyrophosphate. This first E1 is composed of thymine pyrophosphate thymine is vitamin B1, right? The second component is composed of two coenzymes. It is composed of two coenzymes. One is lipoic acid and the second is coenzyme A. Aftab, now you will, Aftab, what are you doing? It's okay. This is your answer. From where the coenzyme A occurs. That is the component of the E2. That is the second component of E2, coenzyme A. But remember, when an enzyme catalyzes a reaction, it remains in unchanged form in last and will undergoes the same reaction again, right? And this component is also composed of two coenzyme, right? One is FAD plus, second is NAD plus, right? So the whole this complex is composed of five coenzymes, 1, 2, 3, 4, 5, right? And whole this complex is composed of three components, E1, E2, E3, right? Now from here I have the pyruvate, which is 3 carbon compound. For example, this is the pyruvate, which is 3 carbon compound. But remember, every enzyme have an active site to which a substrate binds, right? The active site present here can bind only 2 carbons. It can bind only 2 carbons. So for example, here this is the binding site of this enzyme, which can bind only 2 carbons, right? Now pyruvate is requesting to this enzyme for transporting to matrix, right? But he is telling that I have only the capacity for 2 carbons. You have to lose 1 carbon, right? So pyruvate kick out this 1 carbon and react it with oxygen and form carbon dioxide. So net 1 carbon dioxide is released here, right? And only 2 carbons are remaining here, which attach to this site. What is actually this site is? This is thymine. This is thymine pyrophosphate, which is responsible for taking this acetate from here into here. When this site, for example, this is the site, this is the hand of this E1. When this site is filled, this hand start moving, right? When this hand start moving, it transfer this into, for example, here is the site, or here is the site, and having 2 carbon compound. Now this is 2 carbon compound because 1 carbon is lost here, right? He throws this 2 carbon compound into this E2. This is just the jumping system, right? For example, from here this is the 2 carbon and this is E1 and this is E2. This is like this, right? This active site release this 2 carbon compound into E2. So it also have the active site, because these 2 friends are now very sad because they lost 1 friend. So this E2 is trying to make them happy by giving something, by giving something to these two friends, right? Because here we have active site. What is actually this active site is? Do you know what is actually this site is? This is actually lipoic acid. This is actually lipoic acid, and they have sulfurs here. They have, remember, they have sulfur here. For example, this lipoic acid, it have two sites, LSS. They have two active sites which having sulfur, right? So these two carbons which are coming from the E1 are attached to these sites and start moving like this, right? And here on the way, this coenzyme A is added to this acetate. Due to this E2 enzyme, it gives, because this E2 is trying to make this acetate happy, because they are sad, they have lost one friend, one carbon. By giving coenzyme A, they become happy that we have lost one friend, but we are taking the most cash, right, from E2. So here it lost this 2 carbon is having coenzyme A from this E2, but very bad situation or very bad tragedy occurs with this E2 by giving coenzyme A, because these two friends, these two carbons leave their hydrogen here in lipoic acid. For example, here these are the active sites of lipoic acid, these one, because they are moving, right? When these carbons, for example, these are the 2 carbons, they already have hydrogen, but when they take this coenzyme A from E2, they lost their 2 hydrogen, and now these side have 2 hydrogen, and now this LSS is convert into LSHSH. Are you getting this point? Then this hydrogen, what will happen to this side? This is the product which we want as acetyl coenzyme A. This is our product. The 2 carbons with 1 chain coenzyme A. This is the product which we want, acetyl co A. It will undergo Krebs cycle, but here if you see, this carbon dioxide is released. This is decarboxylation reaction, right? So the name of this enzyme is the actual name of this E1 enzyme is pyruvate dehydrogenase. This is the enzyme pyruvate dehydrogenase. Why it is called complex? Because the two other enzymes are helping with this pyruvate dehydrogenase for making acetyl coenzyme A, because this is only can able to make acetate, not it is acetyl coenzyme A. This coenzyme A is added by this E2 reaction. So the name of this enzyme will be dihydrolefolyl acetyl transferase. What is the actual name of this E2? Dihydrolefolyl acetyl transferases, because it transfer its coenzyme A to acetate. So it is called dihydrolefolyl acetyl transferases enzyme, right? Here, what happened to these FAD plus and NAD plus? These are the 2 hydrogen. When this active site is again converting into this situation, that it will take acetate, other acetate, it lose their 2 hydrogen. It will lose There's two hydrogen, and these two hydrogen were taken by FAD plus and will form FADH2. It will form FADH2, right? And again this FADH2 will lose their two hydrogen again and will convert into FAD plus for other reaction. This hydrogen, there are two hydrogen, remember there are two hydrogen, H plus H, because it lost two hydrogen, H plus H. One of the hydrogen will react with this NAD. It will react with this NAD and it will form NADH. NADH plus one proton. One proton will give to this space, and it will consume in other reactions or in other activities, like for example when acetylcholine is going inside the vesicle, the hydrogen ion will be released. So these are the hydrogen which are using for other activities. But this is the NADH which is formed in this reaction. This is the carbon dioxide which is formed in this reaction, right? And this is the coenzyme A which is added to acetyl group, right? So these are the three enzymes. What is the name of this enzyme? What is the actual name of this enzyme? The name of this enzyme is dihydrolipophile dehydrogenase. This is the third component of pyruvate dehydrogenase complex, right? Pyruvate dehydrogenase complex have three components. One is pyruvate dehydrogenase, second is dihydrolipophile acetyltransferases, and the third one is dihydrolipophile dehydrogenases. The function of this enzyme is to release carbon dioxide. The function of this enzyme to add acetyl coenzyme A. The function of this enzyme to reduce NAD plus to NADH. Right? So now this is clear how this link reaction take place. Right? But these are the three components of this complex. There are two other components. But how this enzyme is regulated, first this is important. How this enzyme is regulated. For example, this is the regulatory site of, these are the regulatory sites of this enzyme, right? First, ATP will negatively regulate, when ATP in the cell will increases, it will inhibit this enzyme and will inhibit this whole pathway, right? Because cell now have enough ATP and there is no need of further Kreb cycles, right? For example, we have a lot of NADH in cell. It will also inhibit this pathway, because when NADH are enough in the cell, there is no need of Kreb cycle, because Kreb cycle is responsible for producing other NADH, and NADH will accumulate in the cell and it will affect the cell. So by high amount of NADH, this pathway is inhibited, right? The next thing, calcium. Calcium is the very best regulator of, it is positive regulator, because now listen carefully. When we have muscle cells, and a lot of calciums are entering into muscle cell, the muscles will start contractions. We have discussed this process in detail. By calcium, the muscles will start contraction, and when the muscle is contracting regularly, it will need energy. So when the calcium amount in the cell will increases, it means that the muscles are working, and when the muscles are working, there is a need of energy, and this pathway will regulate it and will form energy. Got it? The next, we have AMP, or we have ADP. These two things will positively regulate this pathway. when the amount of ADP or in the amount of AMP is increasing in the cell, it's mean that energy is deficient in the cell. So this pathway will regulate it and will form energy, right? The next thing is NAD plus. NAD plus will also positively regulate this pathway, because when NAD plus is increasing in the cell, it's mean that cell is deficient in NADH, and this pathway will regulate it and will form more NADH, right? This is the direct regulation. These whole things are direct regulators. Direct regulation, because they are directly affect on this enzyme. There are two very important other two enzymes, E4 and E5, which are indirectly regulate this pathway. For example, one is standing here. There are two other enzymes which are very important, but they are indirectly regulate these pathways, right? One is called, for example, he is standing. One is standing here, and this is called pyruvate dehydrogenase phosphatase. This enzyme is called pyruvate dehydrogenase phosphatase, right? What he is doing, he inhibit this pyruvate dehydrogenase phosphatase. So when the amount of this enzyme is increases, he will inhibit this pyruvate dehydrogenase and will inhibit this pathway. But the question is, what are the regulator for this enzyme? Because enzymes are always regulated by other things, right? These are the regulator, but when ATP is increases, it will activate this enzyme, and when this enzyme is activated, it will inhibit this enzyme. Are you getting this point? ATP will activate this enzyme, and when this enzyme is activated, it will be inhibited by this enzyme indirectly, right? And when NADH is increases, it will also activate this enzyme, and when this enzyme is activated, it will inhibit this enzyme, right? When calcium is increases, it will inhibit this enzyme. Calcium will inhibit this enzyme, and when this enzyme is inhibited, it will regulate this enzyme, right? All the things are opposite here, because this is indirect regulation, right? The next enzyme, which is very simple enzyme, for example, here right, and she have ears, right? And for example, I am getting this calcium and I am trying to cover this from this enzyme. The name of this enzyme is pyruvate dehydrogenase kinase. This is pyruvate dehydrogenase phosphatase, and this is pyruvate dehydrogenase kinase. How this enzyme inhibit this enzyme? For example, these are the sites for inorganic phosphates, right? This enzyme insert inorganic phosphate in this enzyme, and when this inorganic phosphate are attached to this component of the enzyme, this enzyme will inhibit it. But what is the role of kinase enzyme? I have already asked you. Phosphorylation, the removal of phosphates. So this kinase enzyme, the name of this enzyme is pyruvate dehydrogenase kinase. It will again remove these two inorganic phosphate from pyruvate dehydrogenase enzyme and will activate the enzyme. So the activators for this enzyme A are calcium. She is wearing calcium, right? And when calcium are attached to this enzyme, which is called pyruvate dehydrogenase kinase, it will remove these two inorganic phosphate and will again activate this enzyme, right? So this is the activator of indirect activator of this enzyme. Now here are how many components of this pyruvate dehydrogenase enzyme complex? 5. There are 5 components. The first one is pyruvate dehydrogenase, second is dihydrolipophile acetyltransferase, the third is dihydrolipophile dehydrogenase, and the fourth is pyruvate dehydrogenase phosphatase, and the last one is pyruvate dehydrogenase kinase. There are 5 components of pyruvate dehydrogenase complex, and there are 5 coenzymes are in this complex. One is thymine pyrophosphate, second is lipoic acid, the third is coenzyme A, the fourth is FAD plus, and the fifth is NAD plus. This is the whole story of pyruvate dehydrogenase complex enzyme, right? Now, if oxygen is present abundantly, this enzyme will work and will convert this pyruvate into acetyl coenzyme A, and this acetyl coenzyme A will undergo Krebs cycle, and from Krebs cycle it will form 6 NADH and 2 FADH, and when these two components are entering into electron transport chain, it will convert into ATPs. This is the story when there is oxygen. If there is no oxygen, or lack of oxygen or less oxygen, this pyruvate will undergo the reaction and will form lactic acid. It will form lactic acids, right? Which enzyme is responsible for this reaction? Anyone here? This is lactate dehydrogenase. Like this is pyruvate dehydrogenase. Here is the enzyme lactate dehydrogenase, right? And here the competitive inhibitor for this lactate dehydrogenase enzyme is oxamate. The competitive inhibitor for this is oxamate. Oxamate is present in lot of foods, etc. It will inhibit this step, right? So this is if we have lactic acidosis, we can treat by competitive inhibitors, right? But when this pathway is undergoing again and again this lactic acid, the cell will increase in the amount of lactic acids. What is the normal amount? From 4 to 15 mg per dl, right? When this condition take place, why this condition take place here? There is one heredity mutation, mainly in the males as well as in the females, but this is X chromosome abnormality, X chromosome abnormality. When the genes responsible for making this enzyme, because it's protein and the genes are responsible for transcriptizing this, are the transcriptions of this enzyme. So when that gene is defected in the male, mainly because male have X and Y chromosome, and when one X chromosome, when there is one X chromosome and this one X chromosome is defected, only the genes is present on X chromosome, the male will affected, right? And female have two X chromosome, XX. So when one gene is affected, the other gene is functional, right? When on one chromosome the gene is affected, the other chromosome, on the other chromosome the gene is functional, right? So here, when there is defect in the X chromosome or the gene, for example, this is the cell and it have one X chromosome and one Y chromosome, right? On an X chromosome there are the genes which are responsible for the production of pyruvate dehydrogenase, right? Now if this gene is defected, this pyruvate dehydrogenase will not synthesize because the genes responsible for this production is defected. When this pyruvate dehydrogenase is not synthesizing, this pyruvate cannot convert into acetyl coenzyme A, and the cell will overdose in pyruvates. And when there is no pyruvate dehydrogenase, and when the pyruvate is not converting into acetyl coenzyme A, this pathway will run in this direction and it will form lactic acid, a lot of lactic acids. And when a lot of lactic acid is formed in the cell, it will lead the body to lactic acidosis. Like for example here, if there is more than 15 mg per dl lactic acid in the body, it will cause lactic acidosis, and lactic acidosis is very important clinical feature of this link reaction, right? There are three types of this lactic acidosis, right? This abnormality occur in fetal stages, in zygote formation, when there is crossing over during meiosis, right? So when there is defect, this condition occur inside the uterus. So the first condition is very severe lactic acidosis, and in this case the fetus will die in uterus, and this condition is called intrauterine death. So now I hope you will know why intrauterine deaths occur. This intrauterine deaths occur due to lactic acidosis, right? The second type is moderate lactic acidosis, right? Moderate lactic acidosis, this is more complex than this. For example, if the baby is die in uterus, you will just done and deliver this dead baby and will this is the end, right? But when this baby is delivered, alive and will die after 5, 6 or 7 days, this is not good, right? So in moderate lactic acidosis, the baby die in infancy after delivery, right? And the last one, which is very dangerous, is mild lactic acidosis. This is very dangerous, because in this type episodic ataxia is caused due to high concentration of carbohydrates inside the body, the high concentration of lactic acid inside the body. What is ataxia? There is no control on body movements, right? There is no control on body movements. This is just like when a person drunk alcohol, wines, and they are walking like they haven't control on the movements, also on the talking. So this is that condition. In mild lactic acidosis, the patient doesn't have control on the walking, also on the body movements, right? And this condition is called ataxia, and it occur when it is due to lactic acidosis. It occur in episodes, so it is called episodic ataxia, right? There is no control in movement, right? So these are the clinical points according to this link reaction. And why it occur? It is occur due to this defect in the X chromosome. This is, for example, male, and this X chromosome, this is Y chromosome, right? X chromosome is responsible for the gene which is responsible for the pyruvate dehydrogenase synthesis is present on the X chromosome. Now, for example, we have a female, and she have two X chromosome, right? And this chromosome have also the gene, and this chromosome also have the gene for pyruvate dehydrogenase synthesis, right? When there is defect in the one gene, and there is a defect in the one gene, this other gene is functional, right? And when it is not functioning, it will function. But unfortunately, very unfortunately, when there is lactic acidosis, when one gene is defected, the other gene is inactivated directly, and it is called bar switch opening the gene, Barber gene. This is called Barber gene, right? When one is not working, the other is inactivated directly. This is only in the case of lactic acidosis, and that's why this condition is same in male as well as in female. This is the unfortunate condition in females, right? When one gene is defect in the X chromosome, X1 chromosome, the other gene which is present alternative to this on the other X gene inactivated automatically, right? And will cause lactic acidosis, and all these conditions can be possible in females also, right? If this condition was not, imagine if this gene is not functional and this gene will functional, this condition will not will be present in females. But in some cases, these genes, for example, this gene is activated and this gene is dominant. This is dominant, and the effective gene is recessive. This female is only the carrier of this disease. She is only the carrier of this disease, but she doesn't have lactic acidosis itself. She doesn't have lactic acidosis, but she can transfer, mean that when the babies are coming from this female which have one recessive gene, they have 25% chances that the baby is having lactic acidosis, right? So this is the whole story of linked reaction, and our appetite is also depends upon this reaction, because here is a thymine, here is the thymine, and when this thymine is properly working, this pathway will running, and we will need more food to get more energy. But when we cannot eat, right, the doctors are prescribing B complex, for example lysovate, right? They are prescribing for recovery of anorexia. Why? Because this B1 enzyme is responsible, B1 vitamin is responsible for removing one carbon from pyruvate, and when one carbon is removed from pyruvate, this pathway will run, because it will convert into acetyl coenzyme A, and acetyl coenzyme A will undergo Krebs cycle and it will form energy, and when this energy is forming, you will need more food for more energy. And when this thymine is not working, this pyruvate will cannot convert into acetyl coenzyme A, and all these pathways will inhibit it, so you will no need more food for getting energy, right? So that's why we are prescribing vitamin B complex in the patient who have anorexia or loss of appetite. ठीक था? Which way? Any question up to here? Any type of question related to this topic? Okay, we will in next lecture we will discuss Krebs cycles in detail, and we'll see how these NADH, 3 NADH, 2 FADH and 2 ATPs, how they are formed in Krebs cycle, right?